How you doing? This is Dan Giganti from Evolve, and I'm here with Brett Averso. Brett, how are you?
Hi, Dan. Doing great. How are you?
Doing fine. So what are we looking at today? What is that?
Yeah, so I have up here a protein, and this protein is the spike glycoprotein. And this sits all over the surface of the coronavirus. And the coronavirus gets its name because of this protein, because it's crown shaped. It's got these ridges at the top, and if you flip it over, you see it's got kind of like this triangular shape when you're looking at it face on. And if you go on Google Images, just type in coronavirus, you'll see these like triangular proteins all over the surface of any illustrations of what the virus looks like.
So Brett, what's so special about it?
Yeah, well, Dan, the spike glycoprotein attaches the virion to the cell membrane by interacting with a host receptor, and that initiates the infection.
Okay, now what do you mean by host receptor?
Yeah, so that's a good question. The host receptor, so we're the hosts, as we know, and the unfortunate hosts. And the host receptor is angiotensin converting enzyme 2. And so actually I could show that. So why don't I'll pivot over and I'll show that right now. So the yellow protein you're looking at is angiotensin converting enzyme 2. That is a protein that sits on the surface of our cells. And sometimes this protein is soluble and it floats around kind of like in our extracellular spaces. But generally you can find this protein throughout our body and most very prevalently in our lungs.
Interesting.
And so this is this is our host protein for the virus, where the virus will dock and start its infection.
And that's obviously why we see COVID-19 affects the lungs.
That would be correct, yeah.
Brett, you said in the previous video that this virus is actually very similar to SARS-1. How does that impact what you're showing us here?
Yeah, so what I put up on the screen here is the latest strain, the coronavirus, overlapped with the SARS-1 coronavirus protein. And remember, these are just segments of the spike glycoprotein, but these are both the segments that are active for the binding. And one thing you can see as I rotate it around is there is a pretty strong degree of structural conservation between SARS-1 and this strain of coronavirus. There is one other strain that's even more conserved, which is a bat form of the coronavirus, which I do not have the crystal structure of. It's important to note that not only is there some strong degree of structural conservation between these two strains, but the entire viral genome of SARS-1 is quite similar to the latest strain of coronavirus. That's one thing, and then I'll show you something else as well. So forgive me if this is overwhelming, but these are 4 different antibodies. They're all different colors. They're the white, the yellowish, lime greenish color, and the beige one. They're all antibody fragments that in research papers have bound with SARS-1.
Okay, now, so can we use AI to make these antibodies, perhaps the ones on the screen, hit this latest strain of coronavirus?
That's a really good question. The answer is that's a question about cross-reactivity. And so there's been a research paper that recently came out on that topic. They found that these antibodies, when docked against the glycoprotein in a wet lab study, they demonstrated a very poor job of binding against the spike glycoprotein, despite all the similarity between these two. And it's also important to note that this present strain of the coronavirus binds with our angiotensin converting enzyme 2, the host receptor that we host. This present strain binds 10 to 20-fold higher than SARS-1, according to a couple research papers. So given those challenges, we can't readily just combine hope for cross-reactivity between the present existing antibodies against SARS-1 with this latest strain. But there is the possibility with what we do here at Evolve with AI to create variants of these antibodies intelligently and attempt to dock these also within the computer and predict their binding affinity and their other important characteristics, and potentially develop a cross-reactive version of these antibodies that could bind with this latest strain of coronavirus. So there is that possibility.
Thanks so much for showing us this, Brett. I think it's always useful to have a visualization to kind of get a better understanding of what's going on. I also think this helps to sort of dispel some of the myths that are surrounding SARS-CoV-2. As we can see from this is actually quite similar to SARS-1, albeit presenting its own challenges, of course.
Thanks very much, Daniel. Thanks for having me on.